Let's look at the basic mechanism of DNA replication now. What's the process and what enzymes are involved? How actually does DNA polymerase work? Our polymerase is DNA dependent. It requires a DNA template as well as deoxyribose nucleotide building blocks. Remember that one end of a strand of DNA ends in a phosphate attached to the 5' carbon of the deoxyribose, and the other end of that strand ends in a sugar with a free hydroxyl group on the 3' carbon of the sugar. So 3' refers to the sugar end of the chain, and 5' refers to the phosphate end. Remember also that these DNA strands are anti-parallel, but on each strand synthesis occurs only from 5' to 3'. Nucleotides are added to the 3' end only. Each new nucleotide is added by a condensation reaction to the 3' end of the growing DNA strand, and the 5' phosphate of the new nucleotide must be added to a free 3' OH group of the sugar of the previously added nucleotide. Hydrolysis of a pyrophosphate off a nucleoside triphosphate provides the energy. How does DNA polymerase begin a new strand from scratch? Well, it doesn't. It requires a primer that has a free 3' OH group on it, which the DNA polymerase can grab onto and extend by adding nucleotides to that 3' end. A primase enzyme, which is actually a DNA-dependent RNA polymerase, generates a short piece of nucleic acid, about a dozen nucleotides long, that is then extended by a DNA polymerase. Another polymerase subsequently replaces the RNA primer with DNA nucleotides. If polymerase can only synthesize DNA in a 5' to 3' direction, how does DNA polymerase make DNA on the other template strand? In other words, how can you get the replication fork moving in one direction, to the right in this case, if you can only synthesize 5' to 3'? Well, on one strand, the initial primer is lengthened continuously as the replication fork grows, but on the other, called the lagging strand, DNA is made discontinuously, still lengthening the 3' end. The result is a series of Okazaki fragments, each made following its own short primer as the fork opens up and exposes a new region of DNA to replicate. The lagging strand is synthesized as small fragments, about 200 to 2,000 bases long, lengthened by DNA polymerase until it hits the previous fragment, but polymerase cannot join two fragments together. At each origin of replication site, the original DNA helix opens and replication enzymes begin their work. Two replication forks are formed, each with a leading strand and a lagging strand, which are in opposite orientation because of the anti-parallel nature of DNA. This animation focuses on one replication fork, showing the primer nucleotides in red. DNA polymerase extends the 3' end of a primer with new nucleotides shown in blue. As more of the helix opens, synthesis on the leading strand is continuous, but synthesis is discontinuous on the lagging strand, and Okazaki fragments are formed. Another problem. At this stage, the lagging strand is just a series of DNA fragments. The RNA primers have been removed and replaced with DNA, but we still need to connect these Okazaki fragments. DNA ligase joins the 5' phosphate of one DNA fragment to the 3' OH end of the next fragment in a condensation reaction. It uses energy from ATP or NAD intermediates and seals the covalent bond. Ligase prefers double-stranded substrates with no gaps where nucleotides are missing. This huge dynamic complex of DNA and protein machinery at the replication fork is called the replisome, and all of those proteins have specific jobs to do. Before the primase and polymerases can get started, however, the double helix must be unwound, open, and stabilized. A helicase is like a molecular motor. It uses chemical energy, ATP, to break the hydrogen bonds between the bases and to unwind the DNA double helix into single strands. However, this leads to another problem. As helicase unwinds DNA, it introduces a supercoiling force that will resist forward progression of the replicating fork. Enzymes called DNA gyrases and topoisomerases break the DNA backbone, releasing the supercoiling. They introduce transient single-stranded or double-stranded breaks that allow DNA topology to change and then rejoin the DNA strands. Imagine the supercoiling in an overwound string and think about extending that to the forces that resist opening the DNA. One last problem to solve. Once the helicase unwinds the DNA, the two strands don't spontaneously stay open, so single-stranded binding proteins stabilize the separate strands, preventing the strands from reannealing and from being degraded. DNA polymerases synthesize DNA within a complex of protein machinery called the replisome. The synthesis of new complementary strands is initiated by a primer on both the leading and the lagging strands. DNA ligase joins the fragments together.